Hey guys, so in this little video, we are revisiting the structure of enzymes. So as you should recall, enzymes are made out of proteins, right? And so these structures are just a bunch of folded amino acids. Right? And the structure of proteins is determined very heavily by the bonding between different amino acids functional groups. And those bonds are not always super strong. So the shape of these enzymes is actually quite fluid, right, as are most proteins, and they can change depending on the conditions of the cell. Right? But the areas that we're most concerned about when talking about enzymes are the regions that are actively engaging with the substrates. Right? And so those regions are called active sites. Right? And they are shaped in a way that is complementary to the substrate that they are going to engage with. Right? They're very specific for very specific molecules. Not only are they shaped in a specific way, but the chemical properties of the functional groups that are exposed within those active sites are also complementary to the substrates. What that means is that these little areas of active sites are very specific and very attractive to very specific substrates. Right? And so in this enzyme that I've designed here, this is my triangle squarase. Right? Enzymes typically end in ASE as a suffix. I have a triangle active site with regions that would be attractive to my triangle substrate, and I have a rectangle active site with regions that would be attractive to my rectangle substrate. And so when the enzyme interacts with these substrates, right? So these substrates are floating around, and the enzymes are floating around within the cellular regions, right? When they bump into the right spot, okay, the triangle will fit into the triangle active site. And when that happens, there are weak bonds formed between the enzyme's active site and the substrate. Right? Typically either a hydrogen bond or an ionic bond, something that will hold that substrate in place for the moment. That bonding then changes slightly the bonding within the enzyme itself, right? So the amino acids that were linked together via other hydrogen bonds have now slightly altered their properties because now they're engaging with the substrate, and that changes the overall structure of the enzyme. So that active site is going to clamp down a little bit on that substrate and hold that substrate in place. Okay? The other active site will grab the rectangle, do the same thing, click down on it, right? And then it's holding both of these substrates in place. Okay. That engagement with both substrates will change the overall structure of the protein again, okay? And it will force the substrates into a good position for the reaction to occur. Right? And so each of these substrates is going to have a certain orientation that is required for the reaction to occur, and this change in protein shape, this change in enzyme shape, is going to induce that orientation. Right? And so this interaction between the substrate and the enzyme and the change in shapes that are induced because of this interaction is referred to as the induced fit model. Right, once upon a time, there was the lock and key model, where the notion was that the substrates would be a very specific shape, the enzyme would be a very specific shape, and they fit together like a lock and a key. Right. A more updated understanding of these molecules takes into account that molecules move, right? They're not in the same shape all the time. They're in a flux state. Right? And the enzyme takes advantage of those flux states. Right? So it takes advantage of the fact that proteins are wiggly and can change shape. Right? And it uses those changes in shape to manipulate these chemical reactions. Yeah? And so after the reaction occurs, the enzyme then goes back to its original shape. The substrates are no longer the substrates, now they are the product, which has a different overall shape and overall property than the original substrates did. So it doesn't fit in the active site anymore the way it did before. So once the enzyme goes back to being open, this guy can float away as the product. Right? The enzyme is now back in its original shape, ready to do another reaction thing. Right? One of the cool things taken into account when we think of the induced fit model is the fact that enzyme activity can actually be regulated in terms of rate or on and offness by other third-party molecules. And so if this is my enzyme to start with, I could have it not functional. Right? So I could have a bunch of these enzymes floating around in my cells, but not doing anything yet, because I don't want it to do its job yet. Maybe the conditions of the cell aren't ready for that enzyme to work. Right? But when the conditions are ready and the cell's like, yo, it's time for you to do your job, enzyme, it can release this other molecule that fits into a different region. It's not an active site, because this is not a substrate. It's just a modification region. And when that molecule clicks into that spot, it slightly alters the shape of the protein, right? And it turns this enzyme on, right? So before it had this little pointy part in that would block the rectangle from getting in here, but I add this guy, changes the shape, now it's a rectangle, my substrate can fit in, and voila, enzyme activity again. Right? And so the important thing to consider in this whole story is the nature of protein shapes, and to consider the fact that protein folding is driven primarily by weak bonds. And so changing shapes of proteins is not hard to do. Sometimes it ruins proteins, but in the case of enzymes, it's very important to the function of the enzyme itself. Because, shape determines function. Always. Bye.